Hi, this is Naina and this video is based on class 11th biology, second chapter biology classification. In this video, I have highlighted all the important points of the latest version of the text. So do see this video till the end and it will surely be beneficial for your NEET as well as 12th exams. Here this will also help you to point out the important points from your NCERT. And if you're sitting for reading your text, it will be taking many days, so it will be a tiring job too. So this video will be surely helping you on your last days of exam. So moving into the chapter biological classification, the first point that comes is Aristotle, who gave the earliest attempt for a more scientific basis of classification and used the morphological characters to classify plants into trees, shrubs and herbs, and animals into those having red blood and those that didn't have red blood. And the point comes is Lichens, who gave the two kingdom classification as plants and animals, but it couldn't distinguish between the eukaryotes or prokaryotes or unicellular and multicellular. Hence it became inadequate to classify various organisms. Hence came the five kingdom classification by Archaea in 1969, giving Monera, Protista, fungi, Plantae and Animalia. And the main criteria for classification was cell structure, body organization, mode of nutrition, reproduction and phylogenetic relationships. This table is important. According to my point of view, I think these 3 points are very important from this table. That is, the cell wall composition of Monera is non-cellulosic, that is polysaccharide with amino acid it contains. Okay. Then the cell wall composition of fungi cell wall is present and it is with chitin and without cellulose. Then the body organization of fungi is multicellular with loose tissue. The five kingdom classification, I have made a flowchart. Let's move on to check that. Okay, the Monera were classified as prokaryotic unicellular and the eukaryotic unicellular were called as Protista, and the eukaryotic multicellular was again classified on basis of the presence of cell wall. So those that those that didn't have the cell wall were called as animals, and those that had the cell wall were again classified on the basis of the ability to perform photosynthesis. So those that could perform photosynthesis was called as plants, and that couldn't do photosynthesis was called as fungi. So moving into the chapter again, the next point that comes is the 3-domain system. That is, the kingdom Monera was classified into 2 domains, and all the eukaryotic kingdoms, that is Protista, fungi, Plantae and Animalia, were grouped as the 3rd domain. Hence it became the 6th kingdom classification. The next important point that comes is kingdom Protista, that has brought together Chlamydomonas and Chlorella, which were earlier placed in plants, that is under algae, those having cell wall, and the amoeba and paramecium that were earlier placed in animal kingdom that lacked cell wall were now placed in Protista. The next point is the phylogenetic. What is phylogenetic? It is based on the evolutionary relationship between organisms. Then the first kingdom, that is the Monera, and bacteria are the sole members of this kingdom, and they are the most abundant microorganisms. And bacteria are grouped on the basis of their shape, that is the spherical coccus, the rod-shaped bacillus, comma-shaped vibrium, the spiral-shaped spirillum. This figure 2.1 is very important. Just have a look into it. Then moving on to the next point, the bacteria structure is very simple, but it is very complex in behavior, and bacteria are the ones that shows the most extensive metabolic diversity. And the mode of nutrition followed by bacteria are photosynthetic autotrophs, chemosynthetic autotrophs, heterotrophs that consist of parasites and saprophytes. The first group that comes under Monera is the Archaea Bacteria, and these lives in the most harsh habitats. Those that are seen in the extreme salty areas are the halophils, in hot springs are the thermoacidophils, in marshy areas the methanogens. And Archaea Bacteria differ from other bacteria in having a different cell wall structure, and they can survive in extreme conditions. And these methanogens, they are present in the gut of the ruminant animals. Moving on to the second group of Monera, that is the Eubacteria. And Eubacteria are also called as the true bacteria, and these are characterized by the presence of a rigid cell wall and, if motile, a flagella. The Cyanobacteria comes under Eubacteria. Cyanobacteria are otherwise called as blue-green algae, and these have chlorophyll A similar to plants. Then colonies of these are surrounded by gelatinous sheet, and these forms blooms in polluted water bodies. And these also has a specialized cell called the heterocyst that can fix atmospheric nitrogen. Example for cyanobacteria are nostoc and anabina. There are also chemosynthetic autotrophic bacteria that recycle nutrients like nitrogen, phosphorus, iron and sulfur. The next point that comes is cholera, typhoid, tetanus, citrus canker are the diseases that are caused by bacteria. They reproduce mainly by fission, and under unfavorable conditions they produce spores, and also reproduce by a sort of sexual reproduction by a primitive type of DNA transfer from one bacterium to the another. A dividing bacterium is being given in the figure 2.3. The important point to note that all the figures in your NCERT is very important, and you must concentrate on these figures. Then the next point that comes is the mycoplasma are also considered as eubacteria, are the organisms that completely lack a cell wall, and they are the smallest living cell, and they can survive without oxygen and are pathogenic in animals and plants. The next kingdom that comes is the kingdom Protista, and these are primarily aquatic and forms a link with plants, animals and fungi. The first group that comes under Protista is the chrysophytes. These include diatoms and golden algae. The example of golden algae is desmids. These are microscopic and floats passively in water currents as planktons. Most of them are photosynthetic. Diatoms, the cell wall form two thin overlapping shells as a soapbox, and these walls are embedded with silica, thus they are called as indestructible, and their deposit in their habitat over a large amount of their deposit for billions of years is referred to as diatomaceous earth. And since this soil that is formed by their cell wall deposition is gritty, these are used in polishing, filtration of oil and syrup. The diatoms are also called as chief producers in the ocean. The second group of Protista is the Dinoflagellates. You can look at the diagrams. Please check into the diagrams, okay? The Dinoflagellates, they are mostly marine and photosynthetic, and their cell wall has stiff cellulosic plates on the outer surface. Have that word, outer surface, stiff cellulosic plates. And these have two flagella, one longitudinally and the other in transversely in a furrow between the wall plates. And red dinoflagellates, that is example is Goniolax, undergo rapid multiplication and makes the sea appear red. That is, it forms red tides in the sea. So this is due to the toxin released by such a large number, and it can kill the marine animals such as fishes. The next is the euglenoids. The third one of Protista is the euglenoids, and they are seen in fresh water and also in stagnant water. Instead of a cell wall, they have a protein-rich layer called the pellicle that makes their body flexible. Pellicle, that's an important point in euglenoids. And another important point is that they are photosynthetic in presence of sunlight, and on deprivation of this sunlight or deprived of sunlight, they behave as heterotrophs. And they have these 2 points are the most important. That is, euglenoids have pigments identical to higher plants, and they have no cell wall, instead have pellicle. Okay, then other points are they have 2 flagella, a short and a long. Example is euglena. Then fourth group is the slime molds, and these are the saprophytic protists, and under suitable conditions they form an aggregation called plasmodium. The aggregation of slime mold is called plasmodium, and under unfavorable condition the plasmodium differentiate and forms fruiting bodies, and these bears spores at their tips, and these spores possess true walls and are extremely resistant under adverse condition. The fifth group of Protista is the Protozoans, and all Protozoans are heterotrophs and are believed to be the primitive relatives of animals. There are various types of Protozoan. The first type is the amoeboid Protozoan. Of course, you know that it is based on the locomotory organ. It is classified. So amoeboid Protozoan means pseudopodia is the locomotory organ. Hence, they capture their prey by putting out their pseudopodia or false feet. And amoeba is an example of it. And marine forms have, like as in amoeba, okay, not an example, sorry. Then in marine forms they have silica shell on their surface, and the example of amoeboid protozoan is the entamoeba, which is a parasite. You will learn what this entamoeba and all in the further chapters of 12th. Then going on to the next type of protozoan, that is a flagellated protozoan. These are neither free living or parasitic and have a flagella, and these causes diseases such as sleeping sickness. That it is also called as African sleeping sickness and is caused by trypanosoma. It's an example of it. Then the third group of protozoan is the ciliated protozoans, and these are aquatic and they have thousands of cilia, and the coordinated movement of these cilia causes the water laden with food to be steered into the gullet. What is this gullet? It is the narrow cavity like to bring the water into its body, taking food from the surroundings. See, you can see in this picture of paramecium the gullet, and there are thousands of cilia around the surface that bring water to it. Okay, the fifth type, isn't it the fifth type? 1, 2, 3, 4. Fourth type of protozoan is the sporozoans. Of course, they doesn't have such locomotory organ, and here the locomotory organ is referred to as spores. And these are the diverse organisms, and these have an infectious spore-like stage in their life cycle. The example is the plasmodium, the malarial parasite that cause malaria. Then the third kingdom is kingdom fungi, and this shows a great diversity in morphology and habitat. And some common examples that we see are white spots in or mustard leaves, the unicellular fungi, example yeast, then the wheat rust causing Puccinia, then the antibiotic that were used, that is the penicillium. These all are the examples of fungi. Then these are known as cosmopolitan, as they occur in air, water, soil, or on animals and plants, etc. And these are fungi are filamentous, consisting of long, slender, thread-like structures called hyphae, and network of hyphae is called as mycelium. And these hyphae may be cenocytic, that is multinucleated, or others have septae or cross walls inside the hyphae. Then the cell wall of fungi is made up of chitin and polysaccharide. Their mode of nutrition can be saprophytic, parasitic, or symbiotic, that is maybe with algae as lichens or with roots of higher plants as mycorrhiza. Then the reproduction may be by vegetative means as fragmentation or fission or budding, and maybe through asexual reproduction by spore formation that can be conidia or sporangiospores or zoospores. In the sexual reproduction, they forms oospores or ascospores or basidiospores. The next is the 3 steps, that is 3 steps in sexual cycle, and it's very important according to your board exam point of view. And the first step in the sexual cycle is the fusion of protoplasm, that is called as plasmogamy. The second is the fusion of 2 nuclei called karyogamy, and the third one is the meiosis in zygote resulting into haploid spores. In some fungi like ascomycetes or basidiomycetes, 2 haploid hyphae after fusing immediately does not result in a diploid cell. Instead, there is an intervening dikaryotic stage, that is n plus n in a cell, that is 2 nuclei per cell, and this is called as dikaryon, and this phase is called as dikaryophase of fungus. Later only the parent nuclei fuses and the cells become diploid, forming fruiting bodies where reduction division occurs and haploid spores are formed. So the next point is that the kingdom of fungi is divided into various classes depending on the morphology of the mycelium, mode of spore formation and fruiting bodies. So the first class is the phycomycetes, and these are found in aquatic habitats and are also called as obligate parasites on plants, and their mycelium is aseptate and cenocytic. In asexual reproduction, they produce zoospores, that is motile, or by aplanospores, which are non-motile, and these are produced endogenously inside the sporangium. And some examples of phycomycetes are mucor, rhizopus, the bread mold, albugo, the parasitic fungi seen on mustard leaves, etc. And the picture of mucor is given in your text, and it's better if you write with a red pen on the side of the picture the names, so that while learning you automatically get used to it. So the second class is the ascomycetes, and these are called as sac fungi, and multicellular example is penicillium, and unicellular, which is an exception, is the yeast Saccharomycetes. Then the mode of nutrition may be saprophytic, that is decompose, or as parasite, or coprophilous, that is growing on dung of cattle. Then their mycelium is branched and septate. Then the asexual spores are conidia, that are exogenously produced on the mycelium on the special mycelium called conidiophores. And the sexual spores are ascospores produced endogenously in the sac-like structure called asci, and asci are arranged on the fruiting body called ascocarp. Then the example for ascomycetes are aspergillus, the figure is given in your text. Then the claviceps and neurospora. And neurospora is used extensively in biochemical and genetic work. And the morels and tuffles are also an example of ascomycetes, and these are edible. Then the third class is the Basidiomycetes, and Basidiomycetes are also called as bracket fungi or puff balls, or are also called as mushrooms. Then these are parasitic. They may be parasitic, example rust and smut. Then the mycelium is branched and septate. The asexual spores are not found, but vegetative reproduction is by fragmentation, and their sex organ are absent, but plasmogamy of 2 vegetative or somatic cells of different strains or genotypes results into a structure that gives rise to basidium, a dikaryotic structure that gives it to a basidium. And this basidium produces 4 basidiospores. That's very important. It produces 4 basidiospores, and these basidiospores are exogenously produced on the basidium, and the fruiting body is called as basidiocarp. Some examples of Basidiomycetes is agaricus, the mushroom, the eustilagos, the smut, fungi, and the Puccinia, the rust fungi. The fourth class is the Deuteromycetes, and these are also called as imperfect fungi because only the asexual or vegetative phase of these fungi are known. When we come into the asexual mode of reproduction, it produces a great confusion that whether to place it in Deuteromycetes or Ascomycetes or Basidiomycetes. Hence, it is called as an imperfect fungi. So, the Deuteromycetes reproduce only by asexual spores known as conidia, and the mycelium is septate and branched, and some are saprophytic or parasitic, and a large number of them are decomposers helping in mineral recycling. And the common examples are Alternaria, Colitotrichum, and Trichoderma. Now moving on to the next kingdom Plantae. They are partially heterotrophic. Only I am elaborating the main points in this. Later on we will have another chapter. So the main points are they are partially heterotrophic. Then there are insectivorous plants or parasitic too. That is, the bladderwort and Venus flytrap are examples of insectivorous plants, and the Cuscuta is an example of parasite. Then their life cycle includes a diploid sporophytic phase and a haploid gametophytic phase that keeps on alternate with each other. Hence, there is a phenomena called alternation of generation. The fifth kingdom is kingdom Animalia, and their food reserve is glycogen or fat, and the mode of nutrition is holozoic, and their higher forms shows an elaborated sensory and neuromotor mechanism. The sexual reproduction is by copulation of male and female followed by embryological development. And the next topic is that in Whittaker's classification, he didn't mention about lichens and some acellular organisms like viruses, viroids, and prions. So moving on to the first one, the viruses. These are not considered as truly living and are characterized by having an inert crystalline structure outside the living cell. And these, when infect a cell, they take over the machinery of the host cell to replicate themselves, killing the host. And the meaning of virus was venom or poisonous fluid, and it was first given by Louis Pasteur. And this virus was discovered by various scientists, and the first one was in 1892 by D.J. Ivanovsky, that he recognized certain microbes as casual organism of the mosaic disease of tobacco plant that was smaller than bacteria and could pass through the bacteria-proof filter. Then in 1898, M.W. Bajorinek extracted this infected plant of tobacco. That is, the extract of the infected plant of tobacco could cause infection in healthy plants, and he called this extract as contagium vivum fluidum, that means infectious living fluid. Then in 1935, W.M. Stanley, who showed that viruses could be crystallized, and these crystals consist largely of proteins, and it was inert, that is, these viruses were inert outside their specific host cell. And he also said that viruses are obligate parasites. Moving on to the next point of virus, that is, a virus is a nucleoprotein and the genetic material is infectious. It's a very important point that the genetic material of virus is infectious. Then the viruses infecting plants have single-stranded RNA, and that infect animals have neither single or double-stranded RNA or double-stranded DNA. Then the bacterial viruses or bacteriophage, the virus that infect the bacteria are called as bacteriophage, are usually double-stranded DNA virus. You can see the figure of bacteriophage and the tobacco mosaic virus, and it's very important to note these figures. Then the next point is that the protein coat of virus is called capsid, and it is made up of small subunits called capsomeres that protects the nucleic acid. Then some common viral diseases are mumps, smallpox, herpes, influenza, AIDS, etc. In plants, the symptoms can be mosaic formation, leaf rolling and curling, or yellowing and vein clearing, or dwarfing and standard growth. Then the second one was viroids. It was in 1971 that Teodina discovered a new infectious agent that was smaller than virus and could cause potato spindle tuber disease. And these were found to be of free RNA and lacked protein coat. Hence, they were called as viroids, and the RNA of viroids were of low molecular weight. Then the third one was the prions, and these were consisting of abnormally folded protein, and they were similar in size to viruses, and this caused certain infectious neurological diseases. And the diseases that were commonly caused by prions are the bovine spongiform encephalopathy, that is BSE, commonly called as the mad cow disease in cattle, and an analogous variant, that is the CJD, Jacob disease, which was caused in humans. Now let's move on to the last topic of the chapter, the lichens. This consists of an algal component known as the phycobiont and a fungal component known as mycobiont. And it is a mutualistic association where the algae prepares food for the fungi, and the fungi provides shelter and absorb mineral, nutrients and water for its partner. And these are considered as a good pollution indicators, as they do not grow in polluted areas. By this, we conclude the chapter biological classification. And if you like this video, please don't forget to give your like and do subscribe to my channel for more videos. I shall be updating more videos in coming days, so stay tuned. So thank you. Bye bye.